Okay, so let's solve some multiple choice question to test your learning. I'm not a big fan of multiple choice questions, but I think under the current situation, this is the best way to test your learning and get some feedback on your understanding of the topics. As it is a recorded video, I have the privilege to explain all the options and then justify the right answer. Now, first question, what is the advantage of G proteins tethered to the plasma membrane? As you know that previously I mentioned that of the three trimeric G proteins, G alpha and G gamma are attached to the membrane. And this attachment to the membrane is facilitated by a post-translational modification of the G proteins in which a lipid molecule is covalently attached to the protein. And you know that the plasma membrane is a bilipid layer. So if a protein has a lipid molecule attached to it, it then gets easily attached to the membrane, and that facilitates its movement across the membrane. Now, during G protein coupled receptor signaling, when the ligand binds to the GPCR, there is a conformational change and the trimeric G proteins are then recruited to the receptor. The association of the G proteins with the membrane facilitates its binding to the activated receptor. I also discussed that following binding to the ligand bound receptor, the G protein gets activated and the GTP binds to G alpha protein, which then gets activated and bind to the adenylyl cyclase. And adenylyl cyclase is an enzyme which is also attached to the membrane. So therefore, all these proteins being attached to the membrane increases the chance of finding each other quickly. So that facilitates the signal transduction process very fast, in fact less than a milliseconds. So this is a valid option. The second option, that it facilitates interaction with ion channels. Now this I haven't yet discussed, but there is also under this GPCR signal transduction pathway or mechanism, the activated G beta gamma can also bind to some membrane bound ion channels and activate the ion channels, which then facilitate movement of ions across the membrane, and that creates a membrane potential or electrical impulse. So this is also a valid option because both ion channels and G proteins being attached to the membrane facilitates their finding to each other. The option C is that it facilitates interaction between individual G proteins. Now this is also another valid option because if the trimeric proteins are attached to the membrane, then also that facilitates their association into a protein complex. So the right answer to this multiple choice question is all of the above, that is D. Now the second question is that why cyclic AMP is called a second messenger? Now as you know that the cyclic AMP is generated when adenyl cyclase gets activated following activated G protein binding, G alpha protein binding. So the first option provided here is because it is generated from GTP. Now cyclic AMP is not generated from GTP. Cyclic AMP is generated from ATP. So therefore this is not a valid option. Second option is that because it is generated as a molecule carrying message following activation of GPCR by an external stimulus which serves as a first messenger. Now this is a valid option because the first messenger can be a ligand. When the ligand binds to the receptor gets activated and that recruits the trimeric G proteins to adenylyl cyclase, and then that gets activated and catalytically convert ATP to cyclic AMP. So cyclic AMP acts as a second messenger because it can bind to several effector proteins or kinases and then activate those proteins, which then catalyzes other functions. So this option B is a valid option. Number C is because it binds to protein kinase A. Now cyclic AMP do binds to protein kinase A, and therefore this is also a partially valid option, but the right option is option number B. Now I like to discuss how GPCR was discovered. A historical perspective of any subject field gives a better understanding of the subject. Earl Sutherland was awarded Nobel Prize in 1971 for his discoveries concerning the mechanism of action of hormones. Sutherland and several other scientists working during that period led to the discovery of adenylyl cyclase mediated generation of cyclic AMP, which is a starting point towards our understanding of GPCR signal transduction. Now, Sutherland was an endocrinologist, and he was investigating how hormones work. Sutherland investigated the mechanism by which epinephrine regulates the degradation of glycogen to glucose in the liver. He discovered that epinephrine acts by activating a phosphorylase, which leads to the formation of glucose from glycogen. Around 1960, Sutherland showed that catabolic hormones such as epinephrine, glucagon, and adrenocorticotropic hormone, they induce lipolysis by using cyclic AMP as a second messenger. Sutherland found that this took place by the way of an enzyme called adenylyl cyclase. Thus, according to Sutherland's scheme, epinephrine binds to a cell surface receptor, which then stimulates adenylyl cyclase, causing the formation of cyclic AMP, which then exerts its effect in the cell by activating a phosphorylase. Now, this general hypothesis was first met with a strong criticism by scientists because it seemed to be impossible that a single substance could lead to numerous effects caused by different hormones. For example, all these hormones, epinephrine, luteinizing hormone, adrenocorticotropic hormone, thyroid stimulating hormone, and glucagon, they have different metabolic effects, but all of them seem to be activating adenylyl cyclase and generating cyclic AMP. However, eventually it was shown that a large number of polypeptide hormones do exert their effects by the way of cyclic AMP. But at that time, nothing more was known about this system. Even the requirement of GTP was not known at that point of time. Alfred Gilman and Martin Rodbell, they received Nobel Prize in Physiology and Medicine in 1994 for their discovery of G proteins and the role of these proteins in signal transduction. Now, they discovered G protein while studying the stimulation of adenylyl cyclase by hormones in isolated fat cells and fat cell ghost. The fat cell ghosts are a subcellular membrane fraction resembling sacs of plasma membrane derived from free fat cells, and they were devoid of nucleus. The spherical ghosts were essentially devoid of fat, and they contain 25% of mitochondria, some glycolytic enzymes, and adenylyl cyclase. The plasma membrane of ghosts were semi-permeable to ions and sucrose, and thus they retained some of the characteristics of permeability shown by the plasma membrane of intact fat cells. Now, the advantage of using this kind of nucleus-free subcellular plasma membrane fraction was that you can eliminate those proteins which are not associated with the signal transduction machinery, and thus you can reduce the background signal. Now, Rodbell and Gilman's group found that GTP led to a robust stimulation of adenylyl cyclase mediated production of cyclic AMP in the presence of glucagon, which they termed as a GTP effect. Now, based on this observation, they concluded that a GTP binding protein is mediating the activation of adenylyl cyclase and the production of cyclic AMP. So, subsequently, they used a Cephadex column attached to GTP, and they pulled down the G proteins from the membrane fraction, and they identified a 5 kilodalton G gamma protein, a 35 kilodalton G beta protein, and 40 to 42 kilodalton G alpha proteins. Interestingly, they found that glucagon, vasopressin, luteinizing hormone, follicle stimulating hormone, thyroid stimulating hormone, adrenocorticotropic hormone, and epinephrine, they all increased the production of cyclic AMP by stimulating adenylyl cyclase through the involvement of a specific G-alpha protein, which was 42 kilodalton, and they designated this protein as G-alpha S, or S stands for stimulatory. In contrast, they found that opioids, somatostatin, angiotensin II, acetylcholine, and epinephrine, these factors inhibited cyclic AMP production by inhibiting adenylyl cyclase enzymatic activity due to the involvement of a specific subset of G-alpha protein, which they designated as inhibitory G-alpha or GI. They also found that the involvement of stimulatory GS or inhibitory GI was tissue specific and cell type specific. Now, I like to point out that at that time there was a lot of bias due to the involvement of stimulatory effect and inhibitory effect. So this bias led to some kind of interesting cartoons at that period of time. A cartoon from 1970 drawn by Dr. Pierre D. Maiter suggested that not all investigators could readily observe the requirement for GTP in transmembrane signaling. So in this cartoon, actually, it's very funny because they are saying that, and as you can see, GTP has a strong effect on the standard deviation. Now, this standard deviation is because of the true extremities, like one very stimulatory and the other very inhibitory. So that actually increase the range of data, and that made to this kind of funny cartoons, which I find very interesting.